Last class we were talking about epigenetics, right? Chemical modifications to DNA and the proteins that DNA are wrapped around that affect whether or not genes get transcribed or not. Gene expression. Today we're back to start finishing up this concept. Last time we talked about X chromosome inactivation, where female body is only one X chromosome and every cell randomly gets inactivated, and that's what leads to calico cat patterning, for example. Today we're going to go through an example whether or not epigenetic marks, whether or not methylation, acetylation of DNA and histone proteins might have something to do with sexual orientation. And then I'll leave you with a thought to consider about asthma. Next class we'll go through a case study about whether or not the air you breathe impacts whether or not you get asthma through histone modification, DNA methylation. So these were the main points from last class. X chromosome inactivation. The trend is, if there's any trend at all, DNA that gets methylated turns off gene expression. Why was that? Steric hindrance. So if you change the shape of the C in CpG dinucleotides, right? It was C's followed by G's. Those C's get methylated, methylcytosine. And that change in the shape of the nucleotide can actually affect whether or not proteins can bind to the promoter region of genes and transcribe them. Likewise, reduced acetylation of histones also corresponds with inactivation. Or, in other words, high acetylation is correlated with activation. You can think about it either way. Low acetylation is inactivation, high acetylation is equivalent to genes that are active. All right. I always like to start off this discussion by informing you all, in case you weren't aware, that there is no single A gene. There is not one genetic variant, one mutation, one gene, one allele that is perfectly correlated with sexual preference, heterosexuality versus homosexuality, at least in humans. There have been so many studies done of this that if there had been a single gene, it would have been found by now. So if there's any fact in genetics ever, this is it. At the very least, sexual preference does not have a simple one-gene genetic basis. That led scientists to start wondering, well, maybe it's multiple genes altogether that affect sexual preference, which is more likely than a single gene. That there would have to be multiple changes from someone who's heterosexual to someone who's homosexual or vice versa. There might be multiple genetic differences that all together combine to produce a behavior or a preference. Then on top of that, scientists started to think, well, even beyond complex traits, multiple genes controlling a phenotype. Could there be something about your life history, that is, what's happened to you from birth to adulthood, epigenetics, turning genes on and off that might affect sexual preference? So there are a couple of interesting findings that predate what I'm going to tell you now. I'm going to show you some data from a recent study. But a couple of the older studies that started people thinking along these lines were, first, there was this study a while ago, right, 2001, decade and a half ago, the fraternal birth order effect. If you happen to have multiple brothers, every additional son that a mom has a 33% higher chance of being homosexual. Why would that be? Is there a biological basis to the idea of a fraternal birth order effect? The more older brothers you have, the more likely you are to be homosexual, if you're a male. Thoughts? Does this have any basis in reality, or is this just a study that found some random correlation? What do you think? No comments? Yeah.
Didn't it have something to do with the mother releasing more estrogen in the later births?
It could have something to do with how the mother's, certainly has to do with something about how the mother's body changes over time.
Yeah.
with exposure to more male tissue in her body. That was the story that these authors were telling, basically.
Yeah, 'cause I
So,
remember reading.
yeah, so it's not necessarily proven, but the concept was that the more male babies a mother has carried, the more her body reacts to having XY tissue in her body, and maybe that changes estrogen levels or something else in the body that affects sexual preference in subsequent sons. Maybe. It's a plausible story, at least. I'm not sure yet if it's true. Then, even better for geneticists, not only are we studying siblings, which are somewhat genetically similar, that's okay, brother, 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 all fertilized by different sperm and different eggs. So that's kind of a controlled genetic study, but then you get to twin studies, identical twin studies. So if you take two individuals that are genetically identical, are they always homosexual? If one twin is gay, is the other twin gay? What's that? What does it say? About 20% chance. So does that argue against genes or environment playing a role? If you have identical twins and there's a 20% chance that both have the same sexual preference, is it genes or is it environment more that causes sexual preference? 20%, they're genetically identical, 20% chance that they have the same sexual preference tells us what about the basis of sexual preference? Is it genes or is it environment? It's 20% when you're genetically identical speaks strongly for the environment. Right? It seems like most of the time, in twins at least, when you have two genetically identical individuals, there must be differences in their lives. The environment has nothing to do with genetics that causes sexual preference. So this raises, to prepare you for what's going to come next, the study that I'm really interested to tell you about, we've got a quick video on epigenetics in twins to prime you. And although this looks like it was made for an elementary school class, it was actually a movie designed for college classes. So never mind the animations. It's a useful video.
Identical twins are genetic carbon copies. The DNA sequences of their genes are exactly the same. Yet physically, identical twins become increasingly different over time. Why is this so? Genes provide the instructions for the development and maintenance of the body. Yet a second set of instructions, known as the epigenome, interacts with
Chip and Mark Kelly, identical twins who happen to be astronauts. So a really famous twin study has just begun on these two brothers. What do they do? Does anybody know about what happened? Yeah. Which one? Oh, it doesn't matter which one. This is pop quiz. Which twin was it they sent into space? Right. One of these two, they're both astronauts. One of them was the International Space Station commander, or at least the US version of this International Space Station commander for over a year, at least a year. He just came back to Earth after a year in space early in March. He returned from the International Space Station. Right. He grew 2 inches. But that has nothing to do with genetics because that's just what happens when your spine decompresses when you're in space and there's no gravity for a year. So he came back to Earth. He's 2 inches taller than his twin brother. There's a difference. Is it genetic or epigenetic? Neither, right. It's physics. But what these scientists were looking for now are, they want to know, in the year that the commander was in space, how did his DNA change, if at all, from his twin? More importantly, did his epigenome change? So they're going to be sequencing both of their sets of DNA to find out, were they the same before one of them went into space for a year? What happened when they came back? What happened when Scott came back compared to his twin brother? And more interestingly, what happened to those epigenetic tags? Did life in space for a year cause some genes to be turned off by DNA methylation, histone acetylation? Right? How does life in space affect our genes and gene expression? There's a really cool genetic study that's a twin study that is just now about to start coming to fruition because we now have our biological sample, Commander Kelly, back on Earth, and they can start collecting more of his DNA and start looking at these patterns. So that's an example of a twin study related to epigenetic marks. Now on to sexual orientation. The Kellys, as far as I know nothing about their sexual orientation. That was just a story about how you can use twins to study epigenetic changes. Presented in 2015 at this meeting, and I'm not sure if this study has been published yet, this got a lot of press. Spit in a cup, I can tell you whether or not you're going to be homosexual. Basically was the point of this. Gay or straight, saliva tests can predict male sexual orientation. These researchers said that they had a diagnostic test that was pretty accurate that would tell you whether or not you had same-sex or different sex preferences. Is it a mutation or is it epigenetics? Why are you guys so quiet? Hungry? What does it say up there in the upper right? Epigenetic tags linked to homosexuality in man. So nothing about genetics, nothing about the DNA sequence of a gene, just whether or not certain genes are turned on or turned off. So here was how this study worked. They took 47 pairs of male twins, so 47 families, they got twice that number of twins, right, 94. In 37 of those pairs, there was one twin who claimed that he was homosexual or said he was self-reported that he was homosexual, and one twin said not. And the other 10 pairs, both twins self-reported as being homosexual. So if you're the geneticist, what do you do to these data? You've got DNA sequences, and you've got the epigenomes of all of these pairs of twins. What would you look for? What are the patterns that these geneticists want to see? If you want to find something that predicts whether or not you're homosexual or heterosexual, what's the pattern? What do you expect to be true about the epigenetic marks in these 10? Yeah. You expect these to have the same, either DNA sequences or, in this case, epigenetic marks. What about when you compare homosexual versus heterosexual? Are those epigenetic? What's going to be true at the gene that causes homosexuality? The fictitious gene that causes homosexuality. Will the epigenetic marks be the same or different? Yeah, different. So that's what they did. They took all of these DNA sequences and epigenomes, the signals of methylation and acetylation patterns, and they found that there were a set of genes, a small number of genes, right, 9 chromosomal regions, who knows how many genes really, were associated 2/3 of the time with sexual preference. In other words, these scientists are essentially saying if they could get the epigenomes, whether or not 9 of your genes are turned on or turned off, there's a specific pattern that 2/3 of the time would accurately predict whether or not you were heterosexual or homosexual. One of those genes was an immune system gene. There was another gene that had a neuronal function. So there's not a lot known about the genes that are in these regions of the chromosomes that are implicated by this study. There are loads of genes in each of these regions. So they don't know yet which specific genes, these are just 2 examples. So there's starting to be some evidence that maybe there's a very complex multiple gene basis of when you turn on certain genes, turn off other genes, maybe that affects sexual orientation. So then you might imagine this got a lot of press. Genetic studies of sexual orientation have been getting a lot of press for decades. And these were a couple of the concerns that were raised by different scientists. And I'd like you to be thinking now, because in a minute I'm going to ask you, what concerns do you have about this study and the interpretation of the data? I haven't given you all of the information, but from what I've said, there should be some thoughts maybe that pop up in your head. I wonder if, why was that pattern true? Think about it for a minute. They were concerned about the number of participants. So is that enough pairs of twins to really get a statistically significant comparison between homosexual pair of twins and a homosexual heterosexual pair of twins. And then the spitting in the cup thing was another concern. So the genes that are in your saliva samples, that is cheek cells that have DNA that make a genotype, don't necessarily have the same epigenetic marks, the same methylation and acetylation patterns that other tissues would. So does it really matter whether your saliva has that gene methylated? Maybe you'd rather care what's happening in the brain, or in the pituitary gland, or somewhere else other than your saliva, your cheeks. So what other points of concern might there be about this study? What might make it more valid, less valid? Critique. Yeah.
Maybe the region they pulled their participants from, part of the country.
Good question. So I didn't tell you that, but that's an excellent question to think about when you see any sort of one of these tests. So where did the participants come from? Is there something in common to your study pool? Are they representative of humanity? Are they representative of a genetically unique population in one area of the country? Multiple races, single races? Yeah, excellent question. So how random are your participants in your study? I mean, what if there's a bias in, do you know if there's a bias in who produces identical twins? Maybe there's something about twinning that predisposes you to also having homosexual twins. Other concerns or thoughts? Yeah.
Twins or multiple siblings being born at the same time don't usually occur until like later a woman's menstrual cycle, usually like
Later in reproduction. Okay, so yeah, if twinning has a correlation with age, maybe it's the age of the mom at birth, not so much that you're a twin, that has something to do with sexual orientation. So maybe epigenetics plays a role later in a mom's life. Other thoughts?
Who's running the study?
Pardon?
Who's running the studies?
Who's running the study? Who funded the study? Are they actually reporting what they found? Do they have an agenda? Is it objective? How were the data analyzed? You implicate the entire study, which is fine. Yeah, something that's important to think about. Anytime you see any sort of statistics, saliva tells you whether or not you're homosexual. Got to look at the details. Other. Yeah. Oh, fantastic. Not that the other points weren't, but one of the key points that I always try to get across to everybody in this class, because I'm trying to produce not only smarter citizens of the world, but also more savvy citizens of the world. Anytime you see something that sounds arbitrary, that should set off alarm bells in your head. Why do they only study male twins? It's sort of an arbitrary decision. They could have easily studied twins of both sexes. So is this true for female twins? Maybe they did both. Let's be skeptical. Maybe they did both studies and they found that there was a correlation only in male twins, not in female twins, so they just didn't tell us that they studied female twins. Would that make you believe the results more or less? Good point. Now to try to tie all of epigenetics back together, remember, we've talked recently about X chromosome inactivation. Moms, and we saw the video at the start of last class, where early in development, each cell will randomly turn off one of the other X chromosome in a female body. And that should be a random pattern. So you get a calico cat that's about 50% orange and about 50% white. Then there was this paper that came out about, again, sexual orientation in males, looking yet for more patterns, not just epigenetic marks in the twins, but is it their mother? Is it the mothers of homosexual, in this case, again, males, not females, they're just looking at males. Is there something about epigenetics in the moms of homosexual males? So in this case, these authors are studying X chromosome skewing. That is, they want to look at the mothers of homosexual men and find out, do their bodies accurately reflect that idea that on average, half of the cells in the mother's body should have one X chromosome active. The other half should have the other X active. What they call skewing then is when the mother's X chromosome inactivation was not 50-50, when most of the mother's body is biased towards having one X chromosome active and not the other one. That's what they call extreme skewing. So in this case, look what happens. When you've got mothers of gay men or mothers of 2+ gay men, what we're looking here on the x-axis is the fraction of cells inactivating the same X chromosome. So we'd expect, what, 50%, which is down here. So women that had gay sons, there were 5 women in this study out of 44 that had gay sons that had that expected 50-50 split of half the X chromosomes were turned off and half their cells, the other X chromosome in the other cells. But if you compare this side to the controls, so here's control mothers, right? Mothers that hadn't presumably produced any hetero or homosexual offspring, male offspring. Only 4 out of 103 women in this trial, 90% or greater of their cells only had one X chromosome turned on. In other words, in 90 to 95+% of their cells, the same X chromosome had been turned off. Skewed X chromosome inactivation. What happens when you look at mothers that have multiple gay sons? How do these graphs change? What happens to that same population? The mothers that have 90% plus of one X chromosome active in all their cells. It goes from something like 4 mothers out of 103. If you've got mothers of 2+ gay sons, you've got like 10 out of 44 mothers. A quarter of the mothers of gay sons now have extreme X chromosome inactivation differences. So there's something, it seems like, according to the study, there's something about whether or not mom's X chromosomes randomly turn off or not. If it's non-random, if there's a bias, then the mom is more likely to have gay sons. That's what these authors conclude. You believe it? Let's have some comments. In particular, what strikes you as arbitrary about this study? Besides the fact that, again, it focuses on males and not females. Yeah.
I thought you just said in the other article that the environment dictates what they become.
That's what those authors concluded, was that, yeah, the epigenetic marks in the sons have something to do with sexual preference. This is not mutually exclusive necessarily. So this is also saying that maybe mom's epigenetics contributes to that as well. Excellent point. So it could be multiple things. It could be genetics plus epigenetics in the moms plus epigenetics in the actual individuals. What else is weird?
If they did like a chi-square test, would it actually be like a good enough reason to say that it's...
Is it statistically significant? So did they do statistical tests, and is this a statistically significant difference? If I remember correctly, it was statistically significant, but here's the real question. Then we've got to move on. What's special about 90 to 95%? I know it was just a class, but can you come up with a reason, a story that you would tell that explains why it would be important that up until 80 or 85% X chromosome inactivation skewing, there's no effect on whether or not you have more or fewer homosexual sons. But as soon as you hit 90%, then there seems to be some sort of a change. There's like a threshold. Mom's cells are randomly inactivating their X chromosomes. Up to 80%, one X chromosome is okay, but if you go to 82%, then there's this magical change. Why would that be? And that's a common concern. Again, this gets back more to creating savvy, both consumers of information and also consumers of products, consumers of hype, right? You should ask, why this threshold? Is it biologically relevant, or did the authors just draw something that happens to be a chance correlation? Speaking of chance correlations, here's the best one of all. This has nothing to do with epigenetics. Trying to make wise citizens of society. Did you know that this was true? Don't look around. Don't look at what everybody's wearing. Women tend to wear red around the time of peak fertility, guys. So these psychologists, scientists, surveyed a number of individuals from, I believe, the UK and also University of British Columbia. I think there were two populations that they surveyed. And they just went out and they polled women, when was the last time you ovulated? And then they looked at them and figured out what they were wearing that day. So you record what's the woman wearing, and then basically without actually asking the women, are you ovulating now? They found out if they were currently in the period of the menstrual period, they wanted to know what they were wearing. And if they weren't, then they were looking for, were you wearing any color other than orange or red? This paper showed a statistically significant correlation between whether or not a woman was menstruating and whether or not she was wearing the color red. Don't buy it. What's weird about this? I haven't given you much information yet, but what's surprising or striking about this? Why should you be skeptical? Drawing parallels here with the epigenetics studies. Statistically significant correlation between ovulation and the color of shirt you choose to wear. Why? What's arbitrary? Does it make sense? Somebody tell me a story why this makes sense. What questions are running through your mind right now? Aside from, when are we going to move on and talk about genetics? What would you like to know about this study? Fantastic point. Did they ask the participants what their favorite color was? No. Was their sample randomly distributed or not? No, they surveyed students that were going to University of British Columbia, or people around University of British Columbia, and from wherever else in the world it was they collected their second set of data from. So maybe those happen to be culturally common colors. Great questions to ask. What else? Yeah.
Did they check to see if there was a game day in that same exact date?
See, exactly. I mean, yeah, was it a game day? Were people wearing the colors of the local sports team or gang? Yeah. No. What else? Why red? Why red? It's not the obvious reason. Why red is the question. These authors had no hypothesis going into this study that women who were menstruating would be wearing red, or that were about to menstruate would be wearing red. So there's a lot of, again, like these twin studies and the sexual orientation studies, there was a lot of post-publication analysis of the data of these authors. And one of the primary concerns was that the authors didn't have a good hypothesis for why that specific color would be the color of choice. So what if they had found that women tended to wear green most often? There was nothing about the color that made a reasonable biological hypothesis. The scientists collected information about what the women were wearing and when they last ovulated. one of the concerns was, well, what aren't the scientists telling us? Kind of like what if the scientists in the twin studies had looked at female twins and male twins and not found significant correlation between the females, so they just didn't tell us about those data? What might these scientists not have told us about their study? What might they have done that they just didn't report when they published the data? I'm not saying they did this, but you have to be skeptical. If you were going to categorize a study based on the color of clothing somebody was wearing, color of shirts, how many different colors would you categorize somebody's wardrobe into, you think, on average? 7 colors of the rainbow, plus black and white, 9. That sounds about 10, maybe, colors. So you go out, you survey some people, you say, okay, this person's wearing taupe, so we're going to call that blue. Right, so you categorize into 10 different colors of shirts, say. What do these scientists do next? We've got 10 sets of data, 10 groups of females that wear different colors. So what was the next thing they did? They said, okay, women that are wearing gray, is there a correlation between that and fertility? No? Okay. Women that are wearing white, correlation between that and time of last ovulation? No. Okay. Black, blue, green, orange, yellow. Red, look, there's a correlation. That was the main concern with a lot of these post-publication analyses, was that when you do multiple comparisons, eventually you're going to find a comparison where there's a correlation. But that doesn't necessarily mean that it's actually true. It's just chance, potentially, that the women they studied wore this color clothing relative to when they had ovulated. So bottom line, more reasons to be really critical. When you ever hear anything like this, if it sounds like there's something arbitrary about the study, or it just doesn't intellectually make sense, why would it be that women would wear red when they ovulate or after they ovulate? It's probably, as Jonathan Gelman said in this article where he picked this study apart, probably too good to be true. Okay. Next time we meet, we're going to talk about asthma and the environment. Because we're in the Central Valley, it's a really useful thing to talk about. Every year, the American Lung Association, these data are last year's, not this year's. This report was just released for this year. The American Lung Association reports the state of the air, or something like that, where they say, in the nation, who's got the worst air quality? And guess what? We're always at the top. We being California, that is. I mean, Fresno County, we're here, we're 5th worst in the nation, but look who's around us. All the other counties around us. Madera, Kern, Kings, Tulare, Fresno, Stanislaus, etc. We have the worst particulate air quality in the nation. Next time we're going to find out whether or not that affects our epigenome. Right? We're being exposed to environmental pollutants all the time. How does that affect us, and does that affect our ability or predilection to get asthma? So I'll talk about next class. From this paper about air pollution and epigenetics. To end class, we've got about 10 minutes left. I want to start thinking about the end of the semester, because it's almost here. So I prepared a short little mock exam. Go to a course, this is for points, but only for participation. So I'm not going to grade this, we will talk about the answers next class. So it's just to start getting you to start recalling information from across the term. Go to course documents on Blackboard. In the URLs folder, there's a link called mock exam. It'll take you to a web survey, several multiple choice questions. So take this and start thinking about recalling information to prepare for taking the final exam. This is for participation points for today, together with the Socrative quiz that we started class with. We've got content Wednesday, Friday, and Monday, new content. Next Wednesday's a review session, and then that's it. So we've got 3 more days of new content, and the review session you should bring questions to for reviewing for the final next Wednesday.